Now in this final video on Unicon Diversity 2, we're just going to be summarizing everything that we talked about in the previous video in terms of the diversification of animals. And in addition, when you look at this final flowchart, which is just going to be words, just going to be names, you can actually go deeper into the details as you move forward with your own studying. And I'll explain the strategy I suggest with this flowchart in just a second. So this is going to be a summary of everything we talked about basically in this whole lecture. And this whole lecture is the idea that we have animals, and animals all came from an ancestral protist. That's our top of our flowchart. This flowchart is going to be a process and an evolutionary history that we're going to show. We're going to show a phylogeny right now. This ancestral protist signifies that all animals have a definite and true single common ancestor, and thus all animals are part of a monophyletic kingdom Animalia. Now, the next big divide that this protist, let's say, the choice that was necessary was whether or not you have true tissues or not true tissues. And that's going to be based off of the fact that whether or not you become a metazoan or do you become a eumetazoan. And now you should be able to differentiate the two. Eumetazoan would be true tissued organism, and a metazoan would be a not true tissue organism. What do they still have? They still have differentiated cells, but eumetazoans have differentiated cells, tissues, and sometimes even organs. And of the metazoans, the one you have to remember is the phylum Porifera, the sponges. The sponges are the exception to every animal rule that we'll establish from this point forward. The only thing that they have that makes them an animal is the fact that they are going to undergo a similar type of animal reproduction, and more specifically, they have differentiated cells, and all animals have differentiated cells, thus they can be considered part of metazoan, and thus they are all animals. They still came from our ancestral protists, thus they are our basal animal. The eumetazoans are going to be further subclassified into the types of symmetry that are seen. Here we have asymmetry, but over here we actually do have a choice in symmetry, and that choice is the following. It's either radial symmetry, which would just be broadly called radiata animals, and also the choice of bilateria animals, bilateral symmetry, two-sided symmetry. In radiata, there are two that you need to know, two phyla of organisms, and those are the Ctenophora and also the Cnidaria. Okay, I'm not putting any descriptions. That's something that the previous flowchart did a pretty good job of. This is just to classify everything and make sure we have a clear picture of what we're understanding in terms of diversification, the diversity of the animals. So we go from metazoans to eumetazoans. Now we have the choice of symmetry, radiata versus bilateria. Within radiata, we have Ctenophora and Cnidaria. Good. In bilateria, we have another subclassification to choose from, and that is the idea of deuterostomia, so second mouth in terms of developmental mode, or the fact that you can be protostomia, first mouth, protostomia. Now, all that we've covered so far, there's more to it than just this, but all that we've covered so far in this lecture is the fact that deuterostomia includes the phylum Chordata, and protostomia includes the phyla, both of these, the Lophotrochozoans. Okay? It includes these guys, and it also includes the Ecdysozoans. Ecdysozoa. Okay? And that is our story. If you can understand this simple summary flowchart and explain why there's a divergence here, why is there a divergence here, why is there a divergence here, and explain the idea behind those divergences, those evolutionary steps that are happening. Every time we create a divide in this flowchart, there's a reason for it. If you can accurately explain why certain things are classified the way that they are, you will truly understand Unicon Diversity 2, and it's something that we're going to be building. This is a very simple, believe it or not, flowchart that's actually going to get a lot more complex in the upcoming lectures.